Okay, so let's go ahead and get started. This talk is sort of labeled EEG, fMRI, and language. And to be completely honest, it's going to cover all of those things, but perhaps not in necessarily the way that you think they're going to be covered. So with that, I'm going to give you guys an overview of EEG ERP techniques. So there's sort of three of these EEG ERP talks. Jen Evans and Slavina and I are all giving them. We kind of divvied up background information across them. So I'm going to give a little bit of an overview of EEG ERP and then some of the uses. I'm going to admit fully that there really aren't a lot of studies doing simultaneous EEG fMRI of language. So instead, I'm going to talk a lot about converging evidence, both in fMRI and EEG. And then I'm going to talk about sort of the importance of doing simultaneous, particularly in language and in reading. And we'll go with that. So I want to take a moment and sort of acknowledge the fact that EEG is the oldest of the imaging techniques, although there are people who claim it won't be imaging. But Hans Berger did the first human EEG in 1924, and later on labeled the alpha wave as sort of the Berger waveform, as a waveform that comes up whenever people are drowsy. And EEG really has its strengths in terms of the temporal resolution. So you've probably seen this, well, if you've watched Peter's talks, you've seen this. If you watched several of the other talks, you've seen this. Whereas you sort of have the time over here and the spatial maps, and you can see that EEG, ERP, and MEG are very, very fine on the temporal accuracy, but not so good on the spatial accuracy, although we're starting to make inroads there where ERP and MEG tend to get better source resolution. Still not quite to the level of functional MRI or even single cell recordings, but there are definitely pros and cons. For anyone who might not be aware, the belief is that EEG is generated in pyramid cells in the brain, and we call these bundles of cells all activating or firing together dipoles. Dipoles then get represented across the human scalp like this, and we can measure the electrical fields on the scalp using electrodes. Here we have a 128-channel EEG net, which is very similar to our setup here at NIH. And using these electrodes, we can monitor your brain waves sort of in real time. EEG has been found to represent or reflect different human brain states. So whether you're excited, relaxed, drowsy, asleep, which so far no one is, that's always a good thing, or in deep sleep. And I used to have coma on here, which looks even slower than this. And unlike fMRI, there is no EEG of a dead salmon, so there is that sort of benefit. If we take the, I'm breaking all the hardware over here. If we take the ongoing EEG and we segment it based on stimulus onsets, we can then average all of those stimuli together and get an event-related potential, which looks like this, where there are different peaks and valleys representing what we believe are different cognitive processes. These first several are auditory brainstem potentials, and then later on we get to these what we call cognitive components, like the N1, P1, N2, and P2. And you'll notice that in this particular plot, the ERP is plotted with the negative amplitude up. Much like driving on the wrong side of the road, I discourage this highly, but nonetheless, you will see a lot of ERP papers with negative plotted up. There's a very long story, and if you want to hear about it, I'd be happy to tell you the story of why negative is plotted up, but for now, we'll just keep on moving. So the evoked potential, or the event-related potential, is a portion of the ongoing EEG, as I said. It's time-locked to the stimulus onset. It gives you very strong temporal information, and importantly, it's incredibly comparable across the lifespan. And here it is in sort of a simplified way. And you'll notice that we label the ERP both in polarity, so positive or negative, and then also the duration or onset of that peak, or just simply the count of that peak. So this is the P70, or it could be the P1, this is the N100, or it could be the N1, the P200, or the P2, so on and so forth. Manny Donchen, somewhere around 1982, made this distinction between exogenous components, these early peaks, and endogenous components. where the belief was that the exogenous components were highly influenced by stimulus characteristics. So if you have a flash of light that is particularly bright or a sound that is particularly loud, then the N1 will be much larger to those louder sounds or brighter images than it would be to softer sounds or softer images. The later endogenous components are thought to reflect internal characteristics of you processing the stimuli. For instance, the P300, discovered by Sam Sutton in the 60s, is reflective of both an attentional component but also an oddball sort of detection where you have a common stimuli like ba, 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 and at some point you throw in a da, and then your brain says, hey, that's something different, processes it. Doesn't matter how loud the stimulus is or how bright it is. This all happens internally. And then, of course, Marta Kutas's N400 in terms of sentences. I'm putting this definition up there mostly for people on the web. It's a sequence, the ERP is a sequence of overlapping components, each perhaps representing activity of different populations of nerve cells, and each sometimes standing in different, perhaps orthogonal relation to experimental variables. That's Manny Donchin in 1979. As I said before, ERPs are incredibly comparable across the lifespan. For instance, this baby pictured here is now, I believe, 26 years old. So aged incredibly well between when this picture was taken and now. You can see that the N1 latency goes down over time, and the P2 latency also goes down over time, and there's sort of a linear relationship and a curvilinear relationship that I'm not going to go into a huge amount. But if we just simply plot the developmental changes of ERPs, you can see that newborns tend to have very soft waves, very slow waves with not a lot of amplitude, and then as we get older, the amplitude gets larger up until a point, and then right around 8 to 10 years of age, the amplitude stops getting larger and the ERPs continue to get a little bit faster, but the amplitude actually starts to go down as you get older, which may mean that we really peak cognitively around the year 8 years of age. I forget when we give up recess, but it's probably related to that. Just as a demonstration, here is a newborn that we're sneaking up on the hospital. So we, of course, obviously get parents' permission before we do this, but this baby is about 24 hours old. Get them young, so they say. The first thing that we do is we measure the head circumference so that we can pick out the right net size to use. The next thing we do is we measure from the nasion to the onion, that little bump in the back of your head, so that we can find the exact center of your head. And then we mark it with a washable marker. And then, like good scientists, we sneak up on our participants from behind and apply the net. Again, this child is about 24 hours old, really doesn't have much of an idea of what's going on. They sort of wake up and they cry a couple times, and then they go back to sleep almost immediately. So this is 128 electrodes. It goes on in the span of about a minute. Next thing we do is we lay the baby back in the bassinets, play, in this case, different, or measure impedances to find the resistance between the electrode and the head, play some speech sounds, and record the event-related potentials. Afterwards, we take off the nuts, dry off the head, sometimes change a diaper, and then rush off to the next child in the sequence before the nurses catch on to what we're doing. I've already talked about ERP nomenclature. Apparently I got ahead of myself. But as I was mentioning before, we have P1, N1, P2, P3, N4. All of these peaks, although they were discovered by maybe happy accidents, have been fairly well characterized. So just to give you an example of the P1, it tends to appear faster in auditory than in visual, 50 milliseconds auditory, 100 milliseconds visual. We tend to know that the scalp amplitude is anterior for auditory and occipital for visual. Hey, that makes a lot of sense. And we have interpretations of all of these peaks. Again, the P1, N1, and sometimes even the P2 are all thought to be sort of driven by the stimulus. The P2 starts to get into attention characteristics. Later on with like the N2, we get into components that you've likely heard about before. Yes?
Question on the infants.
Yes.
So the auditory evoked potential in newborns is a clinical test. You were showing that the response is really, really small. small enough to older? Is it just really small but good enough to be clinical, or is the auditory lobe based a bit more robust than other?
Yeah, so it is good enough to be clinical by a lot. So the average hearing test performed in hospitals is that they basically either put headphones on the child or they put speakers next to the child's head, and if they jerk their head in that direction, then they believe that the infant can hear, and if not, then they believe that the infant might have hearing problems. ERPs are absolutely good enough for clinical testing, and the state of Kentucky years ago was starting to implement EEG as a method for doing hearing screenings. So, yeah.
Is it possible to record all these components from a single electrode?
Yes, it is possible to record almost all of these components from a single electrode, although nowadays it is often cheaper to buy multi-electrode systems than to build your own single electrode system. That said, a lot of these components are often measured with particular paradigms, so you would likely have to pick the right paradigm for a particular component.
And from a single experimental condition?
Not necessarily from a single experimental condition. So, yep. N170 related to face and object recognition. I'm pointing this out because I'm going to bring it up later. It tends to present inferior temporal and is thought to be generated by the fusiform gyrus. In fact, fMRI studies have shown that it's generated by the fusiform gyrus and sort of the inferior occipital, so on and so forth. These slides are all going to be available online, so if you're really interested in these tables, or if you've heard me speak before, I present these in pretty much everything I've done, because they took me hours to make, and I try to reuse them whenever possible. So on and so forth. So out of most ERP studies, what were thought to be the primary interest were the amplitude and latency. So you would measure sort of the amplitude in microvolts of this particular peak and the latency of this peak. So this is a P1 happening at about 125 milliseconds. This is an N2 happening at about 220 milliseconds, so on and so forth. But there's actually a huge amount of additional data that we can get from recording ERPs, particularly when we have more than one channel. For instance, one of the things that has become very popular recently is to look at scalp topographies. So this is a gambling task. Actually, people were playing rock, paper, scissors against the computer. And little did they know that we actually balanced the rate of feedback. So you always won so many times, drew so many times, or lost so many times. These are the things that computers and casinos can do to you. And if you look at the scalp topography, you can see that win and draw have very similar sort of right lateralized positive activation, whereas the lose condition, we should see this medial frontal negativity, which has been often associated with sort of a mismatch between your expectations, because we think we're all winners, and the feedback that you're getting. How could I lose rock, paper, scissors against a computer? How dare they? But it turns out that this actually replicates for all kinds of gambling tasks and any kind of feedback, really. People have also looked at frequency analysis by running FFTs on the data, or sort of the more advanced wavelets, where we can detect both in terms of time and frequency and intensity, or the most recent sort of fad is source analysis, where we start to put things into the same space as fMRI, and we can actually look at the overlap between ERP source analyses and fMRI activation. It's important to note, though, that there is an actual link between amplitude and topography. So if we just, this was a test between infants who were bottle-fed versus infants who were breastfed from birth up until they were about 18 months old, and then we did ERPs on them to look at how they processed language. This was using a particular bottle formula that was very popular at the time. And what we found in this fairly well-controlled study was that infants who were bottle-fed showed sort of much more broad ERPs with slower latencies than infants who were breastfed, and it looks to be represented as sort of a delay in terms of bottle-fed versus breastfed, and that seems to also relate to language processing, or the speed of language processing. Importantly, though, if you compare how the brain responds as an infant, so this is an infant ERP across 128 channels, and we start to see immediately after speech sound sort of this frontal positivity that then goes away, and then we get this sort of posterior left positivity and some negativities here and there. If we compare this to what it looks like in an adult, or even someone who's further along in development or has maybe not a myelination issue, whether it's because normal myelination hasn't kicked in, or someone who has a actual myelination problem. Here's an adult hearing the exact same speech sound, and you can see that the pattern of activation is immensely more dynamic. It moves around. These are exactly the same time courses. One is just an infant and one is an adult. And I know what you're all thinking. So I've slowed this down. This is really over 1 second. Apparently we're having hazardous weather still. So this is a 1 second ERP that I've slowed down to be almost 30 seconds. What you're probably thinking is that, yes, adults hear the sound ba, and they go, huh, that was a sound. Ba, what do I care about it? And then they start thinking about the grocery list. And this is the importance of averaging trials together, because then hopefully you're not thinking about the exact part of your grocery list across multiple trials over and over again. Frequency analysis was a really big deal, still is oftentimes a really big deal, where we can take the ongoing EEG and then we can break it down into different frequency bands that have been labeled delta, theta, alpha, sensory motor rhythm or SMR, beta, and gamma. And all of these have been found to relate to both clinical disorders but also to interesting cognitive components. For example, theta has been found to be highly involved in cognitive control. Whenever you're playing, say, a gambling task and you're really engaged with the task at a time and you're getting that feedback of correct or incorrect, theta tends to show massive bursts of intensity. And recently, gamma has become sort of a hot button issue because it tends to happen at both frequencies above 26 hertz, and most people tended to filter down to about 30 hertz. But now there's sort of an interest in this high gamma that happens almost all the way up to 100 hertz and how that relates to different clinical issues. I'm going to spend the next few slides talking about source analysis because it's oftentimes the place that people want to take it, particularly when you have all of the resources that we do here at NIH for doing fMRI and then putting the two data together. So this is just an example screenshot. What makes source analysis particularly difficult is that you have two fundamental problems. One is called the forward problem, which is, given the sources and a propagation model, what does the scalp topography look like? And then the inverse problem is the opposite of that, obviously. Given the scalp topography and a propagation model, what does the sources look like? A little typo there. I think that typo's been in there for over a year. It turns out that the forward problem is difficult enough because you can model what the head might look like and propagation models of the head by buying, say, human skulls and filling them with jello. This has actually been done. And then putting little electrodes in to inject current into them and seeing how the current flows throughout that jello mold through the skull and possibly scalp. But for the inverse problem, we have basically an infinite number of solutions. So given a particular scalp topography, there are an infinite number of ways that you could have generated that. And what we end up doing is mathematically constraining it, similar to saying, I know exactly where lightning is going to strike in this thunderstorm as long as these mathematical principles hold true. Fortunately for us, oftentimes those mathematical principles give us fairly believable source analyses. But oftentimes the criticism of source analysis was that people were doing it without sort of best practices or things that would make your source analysis more believable. So right now, typically we recommend if you're going to do source analysis, you really need an adequate number of sensors, whether that's 128 or 256 channels like we have here at NIH. You need some kind of accurate head model, which I'll talk about a little bit later on. It's really helpful if you know the geometry of the electrodes in relation to the scalp and skull. So if you actually know where the electrodes are on the head, you might actually be able to map where those currents are coming from. One of the things that has come up a lot recently is whether or not you can know the conductivity of the head. So old models used to believe that the skull was fairly distortive of currents propagating out from the brain. But it turns out that these values weren't necessarily made up, but they were grossly overemphasized. And recent research has found that the skull tends to cause less blurring than we initially feared. So typically what we do is we make do with some kind of propagation model, which gives us a lot of these essentials. But early source models tended to focus on what we call sort of the Stone-Stoke model, where we had concentric circles. The inner one was the brain, then we had CSF, then we had the skull, and then we had the scalp, and we just assigned values of smearing or conduction to each of these. More recent head models have tried to be considerably more accurate by sort of parcellating the skull, the scalp, and the brain into voxels and using these voxels each with their own values of conductivity, and these can even be person-specific. Gives us considerably better source analysis that has been verified using intracranial EEG electrodes and also using fMRI on the same participants. Once we have a particular source model, there are usually two types of source analyses that are done. One is called equivalent current dipole, and one is called distributed linear inverse. This is oftentimes affectionately known as Loretta. So importantly, equivalent current dipole, you assume that there are a very small number of sources in the brain that fire maybe one or two dipoles at a time. And in the distributed linear inverse, we tend to assume that every single voxel or dipole in the brain is active to some extent, but that activity is either higher or lower based on where you tend to see the pattern at the scalp. Not surprisingly, equivalent current dipole, which essentially you place a source in the head, you come up with some nonlinear search for accounting for some proportion of the variance of the scalp topography, and then you kind of rinse and repeat, either adding more sources or trying to better fit your model to account for variance. This method tends to have really high user involvement and low replication, and you have to balance the fact that unlike fMRI, we're dealing with millisecond accuracy. So these sources can change from millisecond to millisecond. So over the course of a 250 millisecond window or 1 second window, you could have a set of dipoles for every single second of that time. Even if you're averaging over 5-second bins, you still have a huge number of possible source models that you might have to deal with over that time. In contrast, the distributed linear inverse, which often goes by the name minimum norm solutions, where we have all of the voxels activated in the head at a particular time, tends to work a lot better because we don't have as much user interaction. But the interesting part of sort of the Loretta models and minimum norm is what we say is give us the solution that has all the voxels in the head active at a particular time, but I want the sum of all of those voxels to be as small as it possibly can be. So give me the minimum activation possible across the entire brain to give me this particular scalp model. And it works incredibly well. Like N170 studies, we get fusiform activation. Gambling studies, we get anterior cingulate. Motor studies, we get motor cortex. It works incredibly well. The problem is that we know that the brain fundamentally doesn't work like this. We know that the brain has particular sets of neurons or sources that fire in response to particular things. So when you get visual stimuli, you should see visual cortex activation and probably less so other activation. All the voxels aren't on sort of at the same time. We also tend to apply weights to this, which really comes down to smoothing, and then regularization, where we try to reduce the effect of random noise, whether that's deep sources. So there's a fundamental belief that EEG doesn't necessarily see all the way down to the thalamus, whereas there's another side of the debate that believes that EEG sees absolutely everything. If you ever get to go to conferences and see people fighting over global signal regression, these people yell louder, and this fight's been going on for longer. To give you sort of the overview, though, in terms of source analysis, you have sort of best, good, and okay. The best is obviously if you have an MRI on every single subject that you have their EEG electrode locations, and you have their, obviously, EEG. Then you can build individualized head models. If you don't have MRIs on everyone, but you still have EEG electrode location and their EEG, you can make do with some kind of a template head model and some custom sensor locations. This will do in a pinch, but obviously it's nice to know where someone's brain is in relation to the skull, in relation to the electrodes themselves. And then sort of I'm going to label it okay, but I would say, probably don't do source analysis on this if you only have EEG and you have no information about where the electrodes are on the head or what the brain looks like inside of the skull. So to get at what the electrodes look like themselves, here's me sporting a very fancy 256 net in this thing that looks like Darth Vader's sort of cocoon that he goes into. And in this we have 11 different cameras that take pictures of me wearing the EEG net. And then using this, we can actually triangulate where all of these electrode locations are and then build a 3D model based on this. Tried to see if I could fit one of these in my office. It turns out it would take up probably the entire office. I'm going to give you just sort of a brief part of this video that just shows, in case anyone's curious, how this works. Again, I'm trying to balance this between telling you about EEG and telling you about what you could do here. So the first thing we do is we mark a couple of these cardinal locations. This is my former graduate student, Monica, working through very quickly how to do this. And once you mark all of these cardinal points, then you get all of the different EEG sensors, and the software kind of takes a guess at where all of the electrodes are in 3D space. But you'll notice that a lot of these electrodes aren't actually the little blue coins or spheres are not actually overlaying with the electrodes themselves. And so what ends up happening is you have to move these little spheres over the actual electrodes themselves. And as you do this, you have to do it in multiple camera angles, but it's sort of like a video game. So you start off with all of these spheres being blue, and then over time, once you confirm them in more than one camera, they start to turn green. We can teach most people how to do this in about 20 to 30 minutes. And at some point, I swear they're going to turn on achievements, and you're going to get little stickers and, like Facebook badges and things like that. But this is a vast improvement from the way we used to do the things where people used to fight not to do this. Now we have people actually fighting to say, gosh, I really want to work on finding the electrodes on people's heads because it's a nice, relaxing afternoon activity. And this continues on for a couple more minutes. This is not Monica in real time, obviously. Okay. So as I said, I'm going to talk a little bit about how EEG and fMRI often give us similar results. There's a plethora of research on reading in EEG and in fMRI, but that said, there is hardly anything written on EEG and fMRI simultaneously and language, which is great because that's how I'm going to make my career. Right? Yeah. Okay. So briefly, there are differences between typically developing individuals and those with reading disabilities and dyslexia. This has been documented in both EEG and fMRI. There are changes in the brain with the development of language, whether you're testing kindergartners before they learn how to read and then second graders after they've learned basics of reading. And then there are changes in the brain following reading intervention where because you went to a reading intervention, you became a better reader, and now that you're a better reader, we see that your ERP or fMRI patterns change. And these findings converge nicely between EEG, fMRI, and also MEG. So for instance, here's ERPs of reading groups. This is 27 children between 9 and 12 years of age. Their ERP amplitudes and latencies decrease as your skill increases. Up here is just we split them into above average, average, and below average readers, and you can see that the correct responses in group is that, as you would expect, the above average readers perform quite well, the average readers somewhat less so, and then the below average readers even more poorly. Particularly interesting is that this is a pattern we see a lot where most people learn how to read words, but as soon as we say sounds like a word or non-word, that's really when things start to break down whenever you ask people to make these sort of lexical decisions. As your reading ability gets better, we start to see hemisphere differences with increased reading. So your left hemisphere tends to become more active while processing language, the more efficient you are at reading. I have no idea why this figure is in black and white. I apologize, but essentially the really high line, or the fast and very high line are your above average readers, and then the others sort of get mixed into the fray. if we look at MEG results, this is Akasimos' group and Andy Papanicolaou, who are both at Houston. And when we talk about listening to words, you can see that in the right hemisphere, there isn't a huge amount of difference between non-impaired and impaired readers. We see a little bit more activation in impaired readers than non-impaired readers in the right hemisphere. But if we look at the left hemisphere, we tend to see kind of an overactivation of the left hemisphere compared to the non-impaired readers just listening to words. The story becomes even more clear if we look at the reading words, where we see in impaired readers here at the top, the overactivation in the right hemisphere and sort of underactivation in the left hemisphere to reading words. And then if we look at non-impaired readers, we see almost exactly the opposite pattern. Hardly any activation in the right hemisphere, lots of nice activation in the left hemisphere. And in fMRI, this is Temple et al., 2001, we see that particularly, these are, sorry, I'm missing some of my labels. You see in the normal reading children, we see some activation in the right hemisphere, but mostly more activation in the left hemisphere. The children diagnosed with dyslexia tend to show more activation in the right hemisphere and sort of underactivation in the left hemisphere. And if we were to just plot from Shaywitz and Shaywitz, with Ken Pugh as the final author in 2002, your reading activation in what's been affectionately known as the visual word form area, you can see that as your Woodcock-Johnson word attack score goes up, so does your activation in this visual word form area. So nice cases for all saying, hey, we can detect whether or not you're a good reader or not so good of a reader in the brain using a variety of different neuroimaging techniques. Intervention is very similar. Here, for instance, is Temple again in 2003, showing that dyslexic children showed increases after remediation in the left hemisphere compared to the right. But what was particularly interesting about this study was that while the normal children showed sort of classic activation while rhyming, the dyslexic children later didn't initially show this pattern, later on show this pattern, but they continue to activate this right hemisphere area. So it's almost like they have these compensatory areas sort of kicking in to be involved in reading that allows them to process language. And keep in mind that these children are actually reading the dyslexics versus the typically developing children. By the end of the study, behaviorally, they're reading at the same level. There's not really a difference. And if we look at sort of just changes in the left temporal parietal activation based on your total language score, you see that there's a nice positive activation. The more improvement you saw in terms of the intervention for reading, the more activation you saw in this temporal parietal area right here, and again right here. Again, Shaywitz and Shaywitz showed that if you just look at year 3 at the end of the intervention minus year 1 at the beginning of the intervention, we see classic areas like inferior frontal gyrus, angular, and visual word form area, which is always encouraging. You become a better reader, you activate the reading circuit more. And then this is actually, I think, one of my favorite slides, even though it's not my dissertation, which is next, where Andy Papanicolaou and Akasimos brought in children before the intervention here on the left and then after the intervention here on the right and did MEG. And what they found was that before the intervention, for the most part, left, right, you see that there is a lot of activation in the right hemisphere in children before the reading intervention, and then following the reading intervention, they tend to show that the pattern of activation has changed. Now we're seeing a lot more left hemisphere activation than right hemisphere activation. And the funny part of this was when this data was first collected, so it's before the time I joined Andy's lab, they really struggled with how to conceptualize it or how to explain it to journals. And so part of the reason why they printed this entire figure is that it's almost the interocular traumatic test of significance. The data literally jumps off the page and hits you between the eyes, and you have to say, yes, I see there is a change here. And then, of course, an ERP study, this came out of part of my dissertation, where we looked at rhyming versus non-rhyming stimuli and found that typically developing children and children who respond to an intervention are fairly different from the children who aren't responding to a given reading intervention. So we added this third group of non-responders. And what was amazing and interesting here that set it apart from some of the MEG studies and the fMRI studies is that the differences between your children who can read at grade level, the responders and the typically developing, happens at about 100 milliseconds. So 100 milliseconds into seeing a word, we see a difference between whether or not you're a proficient reader or not. And then also interestingly is that at 200 milliseconds or so, we see all three groups differentiated. So we see that the typically developing children are different from the responders, are different from the non-responders. So even though they're reading at grade level, there are these compensatory or supplementary sets of information that are really distinguishing these children. And of course, if we look at some kind of classification, like a very simple discriminant function, and we try to classify children based on their ERPs as typically developing responders or non-responders, you can see that we do okay. But if the fundamental question becomes not can we group them into 3 groups, but can we just say whether they're proficient readers or not proficient readers, we see that really we get a 96% classification accuracy off of their ERPs with only one child mislabeled as reading at grade level when they weren't. So I'm going to do that thing that psychologists do often, and since I'm a psychologist, I don't feel bad doing it. General themes are we see activation differences between good and poor readers. We see modulations of parts of the reading circuit based on how proficient you are as a reader, whether it's proficient after a reading intervention or just overall proficiency. We see laterality differences. Typically, we see more activation in the left hemisphere for processing language than in the right hemisphere, particularly if you're a proficient reader. And reading intervention leads to both normalization, that's more activation in the left than in the right, and also, in some cases, compensatory activation, where there are these additional brain regions that are recruited to help these children after reading intervention read at grade level. Okay, now what you probably all showed up for. Yeah?
On all the studies you just discussed, was that single word reading?
They were single word reading or rhyming. They weren't sentence processing. That's like an entirely different voodoo from the single word reading people. Okay. So advantages to simultaneous EEG-fMRI, and here's where I'm really going to stress that the main reason to do simultaneous EEG-fMRI is if your fundamental research question demands it. I wouldn't necessarily say that you have to say, hey, I'm going to put a whole bunch of people in the scanner with the net on them and do my typical EEG or fMRI experiment, unless you really, really want to. But there are certain advantages to doing it simultaneously instead of separately. And the real benefits are that you put people into the exact same environment, which is inside of an fMRI. So it's sort of unfair to say, I had some people saying in a nice tranquil room wearing an EEG cap with the walls painted pretty colors, and then my other group I shoved them into a 60 centimeter bore, and there was this loud banging noise going on the entire time as they were trying to listen to language. That probably distorts how people are processing things just a little bit. You can also look at individual trial modulation of either the ERP or the fMRI bold response, and I'll talk about that a little bit, and how this combination of the information can actually give you interesting, better results because they are very closely tied. And you can look at coherence and resting state relationships at the same time, whether you're interested in default mode network or any of the other networks. You can really start to make mappings between the coherence as we measure in EEG and the fMRI sources, and there is a somewhat of a push, another fairly controversial issue of doing what we call source coherence, where you do source analysis at every single time point and then you map that onto the fMRI. But of course, nothing comes for free, which is that putting people inside the MRI with the EEG cap on causes some artifacts. There are 2 major types. One is the gradient artifact, or MR, I'm just going to call them MR artifacts, which makes your data look like this, which is clearly not your clean ERP. And also the blistocardiogram, or BCG, which is caused by the movement of the electrode within the magnetic field. And in most cases, BCG is going to be referring to your heart beating. And since most people can't stop their heart beating at exact moments, this is sort of unavoidable. But what causes it is your heartbeats and the electrodes, particularly on your face, which is a little bit more fleshy, tend to move slightly inside a magnetic field. Anytime that you have a metal wire inside of a magnetic field, if anyone has a Sonicare toothbrush, it conducts electrical currents, which causes little bits of distortion. Fortunately, very smart people have been thinking about this since about 1997. Gradient artifacts are fairly straightforward to remove. All we have to do is do template subtraction. So we know when the TR markers are because our EEG system is linked to our fMRI system. And we can take our raw signal that looks like this, and we can subtract out the gradient artifacts and get something that looks pretty close to regular EEG. In terms of the BCG artifacts, this tends to be much more prominent on the facial electrodes, as I said before. We know that it follows the heartbeat by almost exactly 250 milliseconds. So what we can do is we can record your ECG or EKG, depending on where you learned that particular term, and we can identify when your heart is beating, and then we can go out 250 milliseconds and use a PCA to model that BCG artifact, and then just like PCA and ICA are great at, you can find the components related to the artifact, throw those away, reconstruct your waveform, and it actually does a fairly decent job of doing this. Two nice examples of studies that have done simultaneous EEG-fMRI. Goldman in 2002 collected just alpha rhythms inside the scanner and using, at the time, fairly creative pulse sequences, and then just tried to correlate the amount of alpha activation with fMRI and found that, in particular, alpha tends to be fairly predictive of negative bold activation inside the scanner. And then I think one of the sort of nicer examples is Debner in 2005. This group did flanker task where if anyone hasn't done a flanker, come see me after this. We're interested in your data. You have a series of arrows, something like this, and then you introduce a middle arrow that is either congruous with the others or incongruous, pointing the other way. And you instruct your participants to respond as quickly and accurately as they possibly can. The cool thing is that people make lots of mistakes. And when you make a big mistake in any task, and we're recording your EEG, your brain gives off what we call an error-related negativity. It happens about 100 milliseconds after the incorrect response. And it tends to be this nice large negativity in the center of the head. And again, this is negative plotted up. We see here's the error versus here are the two correct down here. And if we do just source analysis on this error-related negativity, we see anterior cingulate cortex, which is nice because this matches what we find today in fMRI studies. And then what this group did that was particularly interesting was that they went back to the single trial data, so every single trial of the ERP, and said, was this correct or incorrect? And categorized it, and then took the amplitude of that error-related negativity and correlated it with the bold response. And what they found was that the larger that negativity, the more negative activation you saw in anterior cingulate, which maps on fairly nicely. This anterior cingulate ROI maps on quite nicely to just the straight fMRI results. But they also were very careful in this 2005 paper to say, aha, but there are actually other places in the brain that are active that fMRI is picking up on that ERP may not be picking up on. This used, I believe, a 32-channel system, so now we have 256 channels. If someone wants to rerun this with more channels, maybe we'll find more things. But I think one of the really interesting parts of using simultaneous fMRI EEG is that it actually allows us to do source analysis on individual participants' brains and then push these to group analysis using, say, in this case, free server surfaces using the beach ball transform. And here you can see that we see more fusiform activation in the right hemisphere for faces and more in the left hemisphere for characters. And this is true both in the fMRI results and also in the ERP results, although I couldn't find my image overlaying the two together. But it is a fairly nice overlap. And then, of course, if we look at any of these sort of responses, we can then get the time course. So we can say, where in the brain is something happening that we're seeing significant effects in fMRI? Now give me the time course of that. And here at 170 milliseconds or so, we see that in the left hemisphere, we see a larger activation to character than to faces. So sort of the sky is the limit in terms of the types of things that you can do now with simultaneous EEG fMRI. And hopefully I've given you kind of an overview of all of those things plus language so no one storms me after this. Any questions?
So, with
Is it possible to show two stimuli very quickly so that the EEG responses would overlap?
Yes.
For those two? And then do they add, or how do they?
They definitely distort one another depending on which brain circuits they're interacting with, but it's not a linear combination like it is in bold. Any other questions?
So, can you just outline what you mean by the reading circuit?
Yes.
It looked as if it was angular gyrus, but I wasn't sure.
I normally have an actual slide that does this, and I took out all of my extra slides later on. So typically, it's thought to be composed of 3 different parts. Ken Pugh came up with part of this model in about 2008 is the most recent. Inferior frontal gyrus, which is thought to be related to sort of articulatory recoding. The temporal parietal area, which is thought to do the orthographic processing. So the combination of the IFG and the temporal becomes the dorsal stream, and this is the early on reading circuit that most people end up using, where they go from sounding out words to recognizing words and mapping the words to the actual sounds. And then the visual word form area, which becomes sort of the expertise recognition, where you can look at a word and instantly know what it is. And this becomes the ventral stream, which comes on sort of later in reading. So as you start off reading, most people tend to activate sort of the dorsal stream, IFG, and temporal parietal. And then as they become much, much better, they tend to show less activation up there and more activation in the ventral stream with the VWFA.
So the projections you had, the 2 different lateral projections, the pre and post for the reading intervention.
Yep.
I mean, it looks as if that was angular gyrus, but that.
So this temporal parietal includes angular,
Okay.
yeah, and SMG. Or not SMG, yeah, angular. That'll teach me to cut out my slides right before I give a talk. Any other questions? Okay, well, 3 o'clock on a Friday. Thanks, guys.